Uh, Coming up next, the presentation will be another industry-sponsored lecture sponsored by our platinum sponsor, Guerbet. This presentation will be given by Professor Jeffrey Mackey from University of Colorado. The title of the presentation is Gadolinium-based contrast agent, mechanism of function and molecular design, yesterday and today. Jeff.
Thank you, and it is a pleasure to be here speaking to this prestigious society on the behalf of Guerbet today. What I'm subtitling my talk is that gadolinium will make all clear. I don't know how many of you can recognize what this is, but this is what it looked like the following morning. Of course, Machu Picchu, and this is a spectacular place if you ever have a chance. I had the honor in April. This is a disclaimer, and I do receive some research support and am on a medical advisory board for Guerbet. So here's the objective of this talk today. I have 6 things I want to do. I want to talk about the clinical usefulness and relaxivity of gadolinium-based contrast agents. I want to describe to you how the chelate really defines the safety and the relaxivity or efficacy. I want to be able to compare the different R1 relaxivities and dosing of the currently available contrast agents and discuss how the chelate itself can be specifically and cleverly engineered to increase relaxivity, which is something that's important to me and probably to all of us in this room. And then what I think is going to be of most interest is to talk a little bit more, and I've talked about this before, about R1 and R2 star relaxivities in blood, which is really relevant to what we do in this society. So I don't think it's going to be too hard to convince most people that gadolinium agents are useful. We do 100 MRs per 1,000 inhabitants every year in the USA with 330 million people. We're talking in excess of 33 million MR exams per year. You know, close to 50% of these use gadolinium-based contrast agents, which means we're giving this 10 to 15 million times per year. And there's a reason why, and that is because it really helps us to clinically diagnose pathologic conditions and understand anatomy, of course, including vascular anatomy. So as a 1A, if you will, here, I think that we all understand that gadolinium-based contrast agents are vital to MRA. They're simple, they're mature, they're robust. There's a lot of beautiful other non-contrast techniques. This is a very solid and very used technique. So I'll move on to relaxivity. This is a yellow iguana. You pretty much have to walk around them on the Galapagos Islands. So just to remind everybody what T1 is, if we take our vector, our black vector here, and tip it away from the purple magnetic field, it's going to want to realign. And that time constant is called T1. If T1 is long, this takes a long time. If we do something to enhance the T1 shortening, like a gadolinium agent, this happens very, very quickly. And we define something called the relaxivity as really what I think of as the efficacy of a contrast agent. And it is defined as little r1, which you can think of as being the slope of the curve, which is looking at big R1, which is, of course, 1 over T1 versus the contrast concentration. So the higher the little r1, the more T1 shortening we're going to get for a given contrast concentration. Of course, we can write similar equations relating R2 and big R2 star to little r2 and little r2 star. So how we go about measuring these relaxivities is a little bit tricky, and there's a lot of investigators that have done this. It's done in saline, it's done in plasma, it's done in blood, and that's something I've done a lot with. It's done at multiple field strengths, and then the vendors will come up with a little R1 and a little R2 value that they put in their package inserts to tell you something about how that contrast agent behaves. It's important to note that typically these values are given in plasma, and they're typically measured in a very relatively low concentration between about 0 and 1 millimolar, which is kind of the tissue concentration you're going to see. It's not the blood concentration you're going to see. And the little R2 star is not usually, or to the best of my knowledge at all, put into package inserts, but that's very, very relevant to the gradient echo imaging that we typically do with MRA, and I'm going to talk about that a little bit. So of course, most of the clinically available contrast agents are based on gadolinium, which is a paramagnetic rare earth element that's not native to the human body, has 7 unpaired electrons, and is therefore very effective at T1 shortening, which is really, you can think of it as a catalyst where a gadolinium can relax up to about 1 million protons around it under the right conditions. And the problem is, of course, the gadolinium metallic ion is extremely toxic. So that's where the chelate comes in, and chelate comes from the Greek word chele, which means claw. So the idea is it's kind of holding on to the gadolinium, and the gadolinium is the little purple sphere in the middle with the molecular structure around it being the ligand, and we're going to spend some time talking about that, but that ligand is really what is going to determine a lot of the biophysical and biochemical properties of a contrast agent. And I've listed some of them here. Of course, relaxivity is very important. Now, the different gadolinium formulations all have different chelates, and that's how they differ. And I've written up here some of the primary ways you can read that they do differ, macrocyclic versus linear, et cetera, et cetera. A couple I'll focus on are thermodynamic and kinetic stability, which really relates to the safety of the agent, and then differing degrees of T1 relaxation enhancement, which is, of course, the product that's what we're looking for out of these contrast agents. Now, if you look at the landscape of contrast agents in the US, it's kind of this big, confusing looking picture. All the way on the left, you have the original contrast agent, gadopentate or Magnavist, back in 1988. And one of the reasons I'm here talking to you now is there's a new agent which just got approved. I'm sure you've all heard of it in the US just last year, called gadopiclinol, which goes by the trade names of Elucerim and Vuway. Now, we can make this a little bit simpler because we, as much as in the society we might have loved Ablavar or gadophosphosette, it's off the market. Gadopentate, gadodiamide, gadovercetamide are all group 1 agents, which are more associated with NSF, which we'll briefly touch on, and they are no longer available in the EU, and by and large, not really being used here in the US anymore. So we can get down to really a much more simple looking landscape where there are 6 real agents to worry about. The 4 that I have circled or boxed in a blue box there are all macrocyclic agents, they're all extracellular agents, and the ACR considers all of these agents in group 2, which are the safer agents to use. There are 2 linear agents still on the market in the US, which are Multihance or gadobenate, and Eovist, which is also called Primavist. Multihance is a group 2 agent as well. I think it's an agent a lot of us here have used because of its relaxivity. Eovist is considered group 3, which the FDA kind of says we're not really sure because we haven't used a lot of it. Now, nobody has talked about NSF yet. I think you got to talk about that quickly at any MR meeting. This is a particularly bad case of NSF here, but just to put this into a nutshell, I think we all know almost all cases of NSF came from 3 different contrast agents. I've written what their names are up there. They are all linear agents. They are all group 1 agents. They were all withdrawn from the EU in 2018, and except for Omniscan, which has some limited use still in the United States, they're really not available in the US anymore. The other agents that I've listed here, Prohance, Dotarem, Gadavist, Multihance, and the new agent Elucerim Vuway, there are, a spurious maybe case or 2 here or there, but essentially very, very few cases with either of these. And I want to point out that it's kind of interesting that the ACR put this new agent in as a group 2 agent, which means low risk of NSF, despite the fact we haven't used it a lot, and I think that's based on the fact that it's macrocyclic and a lot of the science and investigations that went into the FDA approval. So my kind of take is that NSF as a radiologist is not really a very big concern as long as we know what we're doing and what contrast agents we use. Now, gadolinium retention is something that's a little bit more of a hot topic. Kanda, who I believe was a Japanese resident, noticed one day that there was increased native T1 signal in deep brain nuclei, and he correlated that with the amount of gadolinium dosages, lifetime dosage these patients had seen, and particularly noted this in some of the linear agents. So that brings me to contrast agent stability, and that's really the kinetics of the binding of the gadolinium to the ligand. And we can talk about a couple of different kinds of stability. We can talk about thermodynamic stability, which means, you know, at the end of the day, how much free gadolinium ion is going to release from that ligand. And then the conditional stability is just this same thing, but brought to a physiologic pH. I think what is really more important and more impactful is what's called the kinetic stability, which means how quickly are we going to reach that equilibrium. And I'll show you a little bit of data here, but this is the first thing I talked about, thermodynamic stability, which is this, you know, equilibrium equation here between the gadolinium and the ligand together versus them being separated. And you can write the K, which is the equilibrium constant, and we talk about a pK, which is a logarithmic. And if the log of K is 18, that would mean there's like 10 to the minus 18th free particles. So it's not very many. These amounts are very, very tiny. But they do differ between the agents. Now, if we look at, you know, two of the agents that are more implicated here, which are, I guess I'm not pointing at those, but OmniScan and OptiMark at the top, they have relatively low, you know, thermodynamic stability, so you might think that's the problem. But if you look below that at Magnavist, it's got a much higher one, and that was implicated as well. A lot of the macrocyclic agents, which are the group 2 agents, are you know, kind of varying in a lot of different places. And you can see that Elucerem, or Viewway, is down at the bottom there, and it's showing up at, should be 15.5. I think you can see what it is there. It's kind of flipped over a little bit, a little formatting problem. Kind of a pedestrian number, but I think that's not really where the big issue is. What the issue is this kinetic stability. So this is data from ID showing that the half-life of this dissociation in macrocyclic agents is measured in hours or days, depending on which agent it is. And gadopiclinol, or Elucerem Viewway, is 480 hours. So it's a very, very long kinetic stability half-life. Whereas when we look at the linear agents, these are all essentially instantaneous. I mean, whatever equilibrium, whatever gadolinium they're going to release, transmetallate, et cetera, happens almost immediately. So I think that this is really the biggest factor in why macrocyclics have less NSF and less retention issues. So to give you an executive summary of gadolinium retention in the Mackey viewpoint here, I mean, we know we see this with all agents, right? So it's not like we don't see it with macrocyclics, we do, but it's just a lot more with linear agents. And the other thing to keep in mind is gadolinium retention is an observation. It's not really a disease. There's no known pathologic consequence, at least yet, that we know about. Nonetheless, the Europeans have decided to eliminate all of the linear agents from the market out of an abundance of caution, with the exception of Multihance and Eovist, which have hepatobiliary indications there. I think we've seen that the linear agents Multihance and Eovist, and to a smaller degree OmniScan, are still on the market, whereas the other ones are off. And I think it's safe to say that there's a very strong push in the radiology and MR imaging world to use macrocyclics worldwide. So that brings me to the next topic, which is relaxivities. So we talked about the fact that gadolinium agents shorten the T1 and the T2 and the T2 star. We can use this, of course, to get more signal and other things. And they do this by enhancing the relaxation of the proteins, as I showed you, or the protons, as I showed you in the little cartoon. And the different formulations, the different ligands, the different brands have different efficacy into how well they do this, which is related to how they're constructed. So this is a table of the US available contrast agents, and I'll kind of highlight here the little r1, And if we note the box of the top 4 of these are what I consider the kind of conventional extracellular agents that kind of are in the 4 range. You know, Gatavist or gatobutrol is a little higher than the others and more like 5. In the middle there is gadobenate or multihance, which has a little bit of protein binding, and we'll talk about that in a moment. And that gives you a little boost in relaxivity, maybe to the 6 to 7 range, which is kind of where Eavist is as well. And the new agent that we're going to talk about here is 12.8, so it's kind of a big bump up, 2 to 3 times what the other ones are. We're going to talk about why that is. And when we talk about relaxivity, what it is the water needs to find and interact with the coordination site on the gadolinium ligand. And this is done, or we can kind of think about this as really, I think about it as accessibility. So there are diffusion factors, there's the kind of the lifetime, the tau m, the lifetime of the water in that inner sphere where it has a chance to interact. There's what's called the tau r, which relates to the tumbling time of the ligand. And it turns out that tumbling is very important to how much access the water can get. If it's tumbling too fast, and the small molecules we tend to use tend to tumble a little too fast, it can cause problems and decrease the amount of relaxivity you might get. And the other thing is the number of coordination sites themselves. So you notice there's a little gray arrow in the middle which says Q1. That means 1 coordination site. Well, you can design a molecule that has 2 coordination sites, which would obviously relax water more efficiently, and that is really what gadopicranol is all about. So let's talk about how you might increase relaxivity. So I mentioned that tau r, you know, for a typical sized chelate that we use is not optimal, and it turns out if you can get that relaxation rate similar to the Larmor frequency, you will be more efficient at relaxing protons. And as I mentioned, these small chelates tumble a little bit more quickly. So what can you do about that if you're designing a molecule? Well, we've dealt with this before, we've seen it. You can bind your chelate to something big, like human serum albumin, or you can just make the chelate itself bigger to try to slow down that tumbling. So the molecule on the left here is gadobenate dimegylamine, which you know is multihance. It has a little side chain on it, which weakly binds protein. That slows the tumbling down a little bit on the molecules that are bound to protein, and that gives us a boost in relaxivity. The agent on the right is no longer available, but that's gadophosphacet or Ablavar, which has a different side chain, which binds protein, particularly human serum albumin, quite a bit more tightly. And we can see what happens here is that these relaxivities go, you know, up from the standard Magnavist, which I put there as a basis for comparison, up to 4, and then we have going to 6, and all the way up to 19. So the Ablavar, because of its protein binding, actually turns into a blood pool agent as well. So really a big boost in R1 relaxivity. Now another way I said we can do this is to make the chelate larger. So this is a multimeric compound that's under phase 3 clinical development right now called gadoquatrine. And what they've done here is they've actually linked 4 macrocyclic chains together to make a much larger molecule to slow its tumbling down. And you can see what happens there. You push this relaxivity up to 11.8. This is another way to do things, is to increase that hydration number, or Q, the number of coordination sites. So this is the gadopicranol molecule. And you can see that there are 2 different sites rather than 1 where water can interact, which is going to give you a large boost in relaxivity. And in addition, they designed this to put on some additional side chains that don't bind protein, but they do have some steric hindrance. These are hydrophilic side chains, and they will slow the tumbling down a little bit. And what you end up with here is a relaxivity, as I mentioned, of about 12.8. So that's 2 to 3 times the agents that we're all used to using, which to me is kind of exciting. All right. So another thing to realize is, we're all used to this dose of 0.1 millimoles per kilo. And most agents, that's the way they're dosed. Gadopiclinol was FDA approved in the US at half that dose, or 0.05, meaning you're going to be giving half the gadolinium if you do it on dose, but getting 2 to 3 times the relaxivity. And that kind of brings up this next point, which is I like to think about the relaxation delivered, which is really the dose times the relaxivity, right? So I've put Magnavist up there as kind of your comparison. If you dose times relaxivity, it comes in at about 4. You can see Multihance is more like 6.3. Elucerem, gadopiclinol, Vuway, even at half dose, is at 6.4. So by giving half the amount, you're really doing at least what you were with Multihance and quite better with anything else. So the delivered relaxivity is the way I like to think of things. And there are some studies that were done as part of the FDA approval here. And on the left is a brain study where we gave, it was gadopiclinol at half dose versus gadobenate or Multihance at full dose. And really the outcome there was things were about the same, which is kind of what you'd expect from delivered relaxivity. On the right-hand side is same dose, so you're giving twice the relaxivity with gadopiclinol because it's got twice the relaxivity at the same dose, and there they saw significantly better lesion contrast, signal to noise, et cetera, et cetera. These are just a couple of examples from the phase 3 study, which we're now done with gadobutrol or Gadovist at full dose versus gadopiclinol at half dose. And, I have a hard time kind of telling the difference between these. They all look pretty similar, which is pretty much what you would expect and pretty much what you saw when they published these 2 papers, both Kuhl and Lovner, recently, saying that gadopiclinol at its FDA-approved half dose was non-inferior, as a matter of fact, to gadobutrol at full dose. As a matter of fact, there was some suggestion that there was some improvements, which you might expect due to that delivered relaxivity, but nothing really achieved statistical significance. This is an MRA done by one of my former colleagues at University of Washington with gadopiclinol at half dose. I think it's kind of very exciting. I think this agent is going to be really a lot of potential for MRA, and we're going to talk about that in just one second, but I want to say that at higher field strengths, at different field strengths, this relaxivity kind of holds up. The blue on the top is gadopiclinol compared against Multihance, Magnavist, Dotarem, and at 1.5T, at 3T, and at 7T, the relaxivity is not falling off very much, and it's maintaining that 2 to 3-fold increase in relaxivity. And this also happens at 0.5T, by the way. All right, here's the part I'm kind of excited about, is the relaxivity in blood. So I've presented this at this meeting before. This is work I did with Greg Wilson a number of years ago, and I kind of told you that little R1 is the slope of that R1 versus contrast, and that's true in plasma. But in blood, as you can see from the original paper, for 3 different agents, this relaxivity falls off, and that is due to what's called the shutter speed effect, which is really the water membrane cycling in the red blood cell. And Greg Wilson very elegantly worked out a numerical solution. So these dark curves you see here aren't just fitting the data. That is the prediction based on these equations, and it works very, very well. So I just repeated this study, brought Greg Wilson down from Seattle, and with the help of Alex Barker as well. This is what we got in plasma for gadopiclinol, a relaxivity of a little higher than they said at 15.2. In blood, the relaxivity did exactly what we expected it to. Using Greg Wilson's equations, it is spot on. It's really quite amazing. So this is very important to understand. The other thing that's very important to understand is little r2 star. What's happening to the T2 star in blood with these agents? So first off, this is going back to that original data that Greg Wilson and I did. The R2 and the R2 star aren't very different in plasma, but they are dramatically different in blood. So you can see how much on the left bottom is 1.5T, the right bottom is 3T. You can see that the little R2 stars go up really, really high at 3T and really, really high in blood. So of course we have to look at that again as well. So on the left there are the 4 different agents we looked at. On the right, left at 1.5T, on the right at 3T, and you can see how high the R2 star relaxivity is in blood. It also happens to be highest for gadopiclinol, which if anyone here does dynamic susceptibility contrast imaging, you might want to take note of that. Could be quite interesting. So how does this, what does this mean for MRA? Well, I know what the R2 star in blood is of all these agents. I know what the R1 is going to be for all these agents at different concentrations. So what I've plotted here is what the signal intensity would be for an MRA, and I think this is kind of exciting, and I hope people can feel similarly. But there's Multihance, which I'm a big fan of. I've always been a big relaxivity fan, but that's the signal intensity you're going to get versus the gadolinium concentration. If we went to this new agent at even half the concentration, we're going to do better. Notice the drop in the curve as you move to the right. That's the T2 star or R2 star effect. This is at 1.5T. At 3T, you've got to really be careful at high concentrations, particularly with this agent, but look at how much more of a signal boost you can potentially get even at its potential half dose. And I've always stood on a soapbox and said, don't inject your contrast too fast for MRAs. This is going to be one where you even have to inject more slowly. So to conclude, I've enjoyed telling you about a new contrast agent I'm very excited about that came out in 2022. I think there are many other contrast agents that are being worked on. Safety is paramount. Macrocyclic is kind of key. A high kinetic stability. We kind of went over the fact that little r1 is that slope of big R1 versus the gadolinium concentration. Of course, the higher that is, the more signal we can get. The available contrasts now range from just about 4 up to almost 13, which is the one I've just told you about. And that increased relaxivity, I think, comes about by what I consider very clever chemistry. And it can be either done with a large molecule with protein binding or with the approach that Guerbet has taken by increasing this hydration number Q. Also, the concept to me that I think really kind of helps with these contrast agents is to think about how much delivered relaxation there is, which is the dose times the relaxivity. And for gadopiclinol, even at half dose, it's at least as much relaxation delivered as the other agents, maybe even a little bit more. And there's nothing to say we couldn't use this agent at even higher dosages if we wanted to. It would be off-label, however. And then the last thing, and just to leave you with, as I mentioned, is that R1, little r1 behavior is sublinear in blood, and it's quite dramatic. But between understanding that and understanding how large the little R2 star is in blood, particularly at 3T, we can really use these equations. They work, they've been validated, and they can really help us to optimize our MRA. So I will leave you with this beautiful sunset in the Amazon, and thank you, SMRA, and thank you to Guerbet for allowing me to do this, and also thank my coworkers, particularly Greg Wilson and Alex Barker. Thank you.
Thank you for this excellent talk. We have time for one quick question, if there's any.
Thanks, Jeff. It's a very clear explanation. Could you speak to an anxiety I have, which is that because we've got a smaller dose, the manufacturers are making that in a smaller volume, which means that we're then having to put in at even slower rates. And in my experience, the slower you go, the more variability you actually have in arm to chest, transit time, etc., etc. And I wonder whether some of that's going to break down and how we would overcome that, whether we have to go to dilute it, for example.
Sure, great question. For a long time, I've done all my MRAs by taking whatever contrast I'm going to use and diluting it up to 40 mLs, because I totally agree. I mean, this particular agent, I think you had better be injecting it at half a cc per second or less. And you're right, if you do that slow an injection, it gets stuck in the arm, it may pop out at some point, you get these heterogeneous boluses, and you don't want that. So I think for MRA purposes, you're probably going to have to dilute this, and I'm sure the pharmaceutical companies don't want to hear that, but that's what we do and what we can do, and I think what we need to do. So, you know, if you were going to use, you know, 20 cc's of Multihance or 10 cc's of Gatavist, or in this case, you know, 10 cc's of gatopiclinol, I would dilute it to 40 and inject it at, you know, 2 cc's a second just to get that flow in there. But I think it's going to be important to work these things out, and that's what I've spent a lot of my time doing, is trying to figure out how to best administer contrast. And we've got all the tools to do it, and it's just slower than you think. That's what you do. Just slow it down.
Great. Let's thank our speaker again.